My name is Kyle Greenland. I'm currently looking at the role of the tricarboxylic acid cycle, the TCA cycle, in DNA methyltransferase activity, and I'm part of the transgenerational epigenetics and evolution team here at the LMS.
My name is Michael. I'm a third-year BMB student, and my project is keeping pluripotency alive. I'm currently placed in the germline and pluripotency group.
Hi, I'm Carmen, and I'm in the functional gene control group. I'm working on three-dimensional chromatin structure.
I'm Elias. I'm in the integrative skeletal physiology group, and I'm working on looking at the age-dependent changes in the bone microenvironment.
Hi, I'm Emily, and I'm working as part of the grants, engagements and communications team. I've been working on and helping out with a variety of different projects, writing articles for the Institute's website and delivering public engagement events with local community groups, doing social media campaigns and doing short little video projects also.
So in second year, we had this stem cell module. I just thought it was really interesting, and I think it was like one of my primary interests at the time. So I thought it would be a perfect opportunity to work in a lab that was specialized in developmental biology and stem cells.
So I picked my project mainly based on the techniques that were involved. So confocal microscopy was something I wanted to work with.
I picked my project because it involved both wet lab and dry lab. So I was able to pick up a bit on everything and learn a variety of skills.
I wanted to experience what working in science communications was like. I've always had an interest in sharing things with other people, whether it's what I've learned or something interesting that I've heard about. And since this project still revolves a lot around science, despite not being a typical lab project, I thought this was the perfect opportunity for me.
The project's really interesting because it has a heavy base in cancer, which is something I'm really interested in. I think I've learned a lot of skills so far, lots of technical skills, so especially new assays I've been able to master.
One skill that I value quite a bit is the organizational skills that I've sort of picked up throughout this project.
I guess I've learned how to integrate into a lab team.
So how to function within the research environment.
I was able to learn how to use a variety of different programs for design, graphics, video editing, and I had the chance to really develop my interpersonal skills and communication skills when I was helping out with these public engagement events.
So in first and second years of lab pod, we only had one day in the lab per week, which meant that it was very stressful. If something went wrong, you'd have to wait a whole week to correct it. And there was a lot of pressure to get things done in that one day. So here you go to work every day, which means that even if something goes wrong during the day, you can have the afternoon to think about it and then you can work on it the next day.
The main difference and sort of what makes this placement really special is the fact that you kind of take ownership of your own project.
What I also find that's really good is that you are working alongside people that have been in the industry for a long time.
One expectation I had that has sort of been disproven is I thought my project would be very minimal, but I've actually felt the research I'm undertaking and the results I'm generating are having a meaningful impact on the objectives and aims of the research group.
I think one of the biggest expectations I had was that it was going to be sort of 9 to 5, really busy.
Some days you'll be busy, other days you'll be much more relaxed, so it's much more flexible than I initially thought.
Because you're so independent and you're kind of working on your own project, you can set your own pace.
So I just kept an open mind with all the things that were given to me to do, because there's so much variety in this role. Probably getting to experience and getting the opportunity to try so many things within the world of science communications and engagement, and also meeting and working with researchers from the Institute and people from local community groups.
A major highlight of my experience so far is definitely the people I've met, so being able to interact with academics who are leaders in their field.
Great to see what sort of ideas they come up with and their work ethic, and I feel like that has a big impact on my experience in this lab placement.
One word of advice I'd have to future students is to definitely pick a project you're passionate about, because there is going to be a point in the project where you start to get frustrated, and that passion is the only thing that's going to carry you through and enable you to succeed at your project.
Check out the PI's blog and the team's website. to try and gauge the research dynamic and what their background is.
And also read some of their most recent papers. It'll really give you a good idea of the techniques they use and the things they do.
Come in with an open mind. Be sort of open to any challenges that you're about to face and not to be sort of afraid of it.
I'd say be ready and willing to learn so many new skills, both personal and practical skills.
And also, don't be afraid to ask questions.
Yeah, don't suffer in silence.